The very good afternoon, all faculty member, researcher, and research scholars, as well as invited speakers gathered in this 5 days online international faculty development program on bridging research and innovation in biomedical science and engineering. Today is the last day of this faculty development program. This FDP is organized by the School of Allied Health Science and School of Biomedical Science in collaboration with the Institute Innovation Council of Golgota University. So we are highly grateful to have assembled a distinguished panel of speakers from India and from abroad who are sharing their expertise, experience, and latest advancement in the field of biomedical engineering and sciences. So this program presents a unique opportunity for all of us to learn from each other, to exchange our idea, and to share your thoughts as and for the discussion. So as a participant, your active engagement and support will play a crucial role in making this program a resounding successful. So we encourage you to actively participate in all the session, ask your query, and share your insight and experience with us. Now I am feeling honored to extend my warm welcome of our keynote speaker, Dr. Nishant Kumar sir, and my senior also. So Dr. Nishant sir has been working as an assistant professor in the Department of Biomedical Engineering at NIT Raipur since 2018, and his expertise is focusing in the field of biomaterials, biomechanics, orthopedics, and dental implant biomechanics and finite element analysis. He has been in the field of teaching and research from the last 8 years. Previously, he had also worked as a various institute of reputed in the India. Some of them are MNIT Allahabad, Prayagraj, Nehu Shillong, and SGSITS Indore. He is handling research projects from different sponsoring agencies as the principal investigator. He is credited with 16 peer-reviewed journals that is indexed in SCI and SCI Extended. 5 books chapter he is in credit, and he has also presented several papers on international and national conferences of repute. Now I would like to invite Dr. Nishant Kumar Singh sir to deliver the lecture on the topic triply periodic minimum surface based porous orthopedic implant. So over to you, sir.
Thank you for introducing me, Dr. Gaurav. And
thank you for accepting our invitation, sir.
Thanks a lot, sir. I will continue this lecture on second laptop, so I will off my camera. That is fine.
Yes, sir. You can, sir. You can, sir.
Okay. Sir, my slide is visible?
Yes, sir. Yes, sir. It is visible, sir.
Okay. So, good afternoon, all. I will be presenting this work, that is triply periodic minimal surface based porous orthopedic implant. So, before going, what is triply periodic minimal surfaces? Like, we will see why and what are the need of these type of porous. In general, solid implants are generally used in orthopedic application, but We have used this method for several advantages that we will discuss later. So before starting the lecture, what is the need of any uh, uh, issue? What is the need of any repair, and what are the populations which needs actual type of bone repair? In this is the data which was collected from several repositories. There are many causes of disability. In that disability condition, bone or musculoskeletal conditions are uh, in a several population. So in that case, like if you talk about what are the musculoskeletal disorders, there are several ones that are shown here, like osteoporosis, injury or accidents like arthritis, and there are rare musculoskeletal condition that may be bone tumors. So in this case of treating any bone repair, we are only concentrated upon on the bone tumor. That is only 15%, but when it occurs, that can cause the morbidity and mortality. Okay, so causes and effects of bone defects. What are the effects when bone defects occur? So there are several results apart from bone defect. That may be bone tumor, that may be trauma, that may be some other pathological complication. There are several type of bone defect by which there are several other treatment methods also. So in that case, joint diseases that are very prominent, that having the reported cases of 343 million in a year, that is osteoarthritis and rheumatoid arthritis. Diseases like osteoporosis and fractures trauma related, that is 436. Spinal disease, that is intervertebral disc degeneration treatment, that having the reported cases of 305 million. That also increases after back pain, and there are several regions of these type of interventions which can be used. So bone tumors, that are amputation, that may be happen, that annual reported cases are 175 million worldwide. So that affects that causes discomfort and pain. Limited motion can happen. It may be bone defect can occur in throughout the body, like in upper limb, lower limb, or any part of skeletal locations. So limited motion, flexibility, and functional strength can happen. Decreased work efficiency. So what is bone defect? Like this is the schematic representation of bone defect. Like that is length exceeding 2.2 and 2.5 times the diameter of affected bone. If any bone diameter, just for example, this bone diameter is 2 to 3 centimeter. If it is defect is exceeding for about 2 to 2.5 times, that is the critical size defect. In such case, there is no possibility of bone regeneration. So, additionally, to treat such type of defects, we have to take some surgical interventions. Accordingly, we have to use some type of implants. So, what are the current implants which are available in the market that are having several issues? So in that sense, we have tried a triply periodic minimal surfaces to modify or to improve the outcome of any type of implants which are used for treating critical size defects. So what are the methods that are these are the methods that can be uh, um, that can be used for masculate induced membrane technique. So the defect can be wrapped with some material. Elzaro method, that is external fixation method, that is also used for treatment of the critical size defect. A scaffold based tissue engineering. So there are several scaffold that are ceramic based also, that are polymer based also, that are metal based also, which are used for the treating these type of defects. And there are metallic bone implants which are used for treating, that is wide worldwide accepted type of treatment method, that is metallic bone implants. And there are autografts, allograft, xenograft, that may be metal based also, that may be ceramic based also, that can be the naturally occurring modified material. There are several type of methods, treatment methods for these type of critical defects. But ultimately, when you go for the outcome of this type of treatment, there are several complications which is coming after some time. It may be late, immediate after the surgery. It may be after 10 days. It may be after 1,000 days. It may be of 10 years. But complications necessarily comes. So, what is the post-operative complications which can occur? That is late infection. That is second one is that is very prominent. That is mechanical failure. So mechanical failure in this case, most prominent cause, that is stiffness mismatch of the bone and implant. The implant which you are putting at particular location of the bone. So there is usually we are using a very high stiffness implant, and bone that having the stiffness which is very low in comparison to that type of implant. In that sense, stress shielding issue arises. Implant weakening also occurs. There are many biological environment which are generating many corrosive environment, many alkaline environment can occur. So these are the prominent reason of implant weakening. That is the second, that is mechanical failure, that is fatigue. So interface bonding, what is the interface bonding? How much interface bonding between the implant surface and the bone surface which is getting after implantation. So for solid implantation, there is lack of adhesion. There adhesion happens, but that is very limited, because it is completely solid. Only that 1 to 5 micrometer of the surface will contribute into the adhesion or the interface bonding. Implant fixation issues, implant loosening can occur post operatively after implantation. It can micro motion can be generated, and implant loosening can occur. So these are the issues which are addressed by incorporating the triply periodic minimal surfaces. So that are the main issues of conventional solid implants. So what are the current approaches to avoid mechanical complication of conventional implant? So that is this figure shows that this is the coating of the implant. So this is the ceramic coating of implant, that is hydroxyapatite coating, which is used into the total hip arthroplasties. So that substantially engross the bone, but still it is not sufficient to get the substantial amount of interfacial bond. In that case, scientists have tried to make the porous implant. So there are several type of porous implant which is available in the universe. So there are many type of porous structure they have tried, but there are very limited application about triply periodic mineral surfaces. So this is another approach, like irrespective of solid implant, scientists are tried using this type of porous implant to correct these type of to improve these type of treatments. So secondly, nowadays, like patient specific implant is also getting popularity. In that sense, the implant that is used on demand, like if any patient having some injury, on demand we are manufacturing according to their anatomical size and location, and we are supplying to the hospital, and that implant is get implanted into the body. So nowadays, this type of patient specific implant is getting popularity. In that sense, we have accounted some modification and some fabrication method and some processing techniques. So how we can expedite this fabrication technique when any need of urgency. So now we'll come up with the porous structure. There are several type of porous structure which is available in the universe. Like for porous material, that is naturally occurring porous structure that you can see also in wood, plant cells, butterfly wings. And man-made, there are many type of that are structural shape, that is stochastic type, that may be foam Voronoi structures, cell topology, that may be open pore or closed pore, or cell orientation, if you'll talk about, that may be the homogeneous, gradient, or heterogeneous. So we have only concentrated upon the, that is periodic structure, that TPMS based porous structures falls in this category only. And lattice base structure, that also gained popularity for several implants, and there are several commercial implants which uses this type of lattice base structure, that is cubic, octet truss, and there are many type of structures which are available, and that are recently being used in several type of implants. But TPMS based implant, that is, for example, squash, P, and diamond structure, gyroid, and IWP structure, and there are many type of TPMS based structure which comes into the or falls into the category of TPMS. So we have only concentrated upon the TPMS based porous structure. Now these are the type of porous structure which we have discussed. So like BCC, diamond, octet, rhombohedric docadron, rhombohedric octahedron. So honeycomb structure, that only can be used into 2D cases only. But TPMS gives many more advantage in respect of these type of structure. So that TPMS is based on the stochastic structure. One is stochastic structure, you can see, that is foam based structure, that mimic the bone morphology, and Voronoi structure, that also mimics the bone morphology, but controlling these type of stochastic structures porosity is very difficult. So we have introduced this TPMS structure. So let's see about what is TPMS, triply periodic minimal surface structure. That is you can also find in nature. That is also a type of nature inspired material, that this type of structure you can also find into butterfly wings, into soap films. That is the surface which minimize the area locally, or possess mean curvature equal to 0, lying on each point of the surface with repeated geometries in 3 directions. So if you'll talk about its surfaces, like this is the type of minimal surface, like mean curvature of any true curve which is plotted upon the surface, that will always be 0 at each and every point. Uh, so why TPMS we are incorporating into the orthopedic application? That is, parameterization of TPMS structure is not uniquely achieved to develop porous implant in an effective way to meet the patient specific needs. So parameterization of TPMS is very difficult. If you talk about the any other type of porous structure like cubic structure, that can be easily parameterized in many CAD softwares. But TPMS based surface structure, that are very difficult to parameterize. Second, biological requirement. Yes. Uh,
Sir, yes, sir.
any
Muted, sir. Muted, sir.
okay.
You can
Okay.
continue, sir.
Sure, sir. Sure, sir. So, biological requirement are there which such as cell attachment in growth, tissue differentiation, proliferation. In that sense, solid implant cannot work. And secondly, when we are using any other type of porous structure, that also having a limited type of cell attachment and ingrowth and differentiation. And there are other many other biological requirement which cannot be fulfilled by the other porous structure. So we try to incorporate this TPMS structure into the implant, so we can achieve these type of functional requirement like biological requirement, parameterization, what is the fluid responses. You cannot achieve the fluid responses in any other implants, like solid implants. If you are using, there is no sense of talking about the fluid. So if you are introducing the intentional pores into the implant, by that you can talk about the fluid responses. Conventionally developed implants that can also be used for large segmental bone defect treatment. Modulus mismatch, that is the most severe problem that it gives a rise of stress shielding, that can also be tunely reduced. Dual approach can also be used for achieving the patient specific implant. On demand virtual post operative approach can also be adopted during the surgical process. These are the advantages which can be achieved when we are introducing the TPMS surfaces. So uh, in this presentation, like I will show, we have several type of challenges which we have removed, like parameterization challenges, biological requirement, fluid responses, anatomical mismatch, that is also achieved with the patient specific condition. Modulus mismatch concerns, like we have reduced the stress shielding. Dual approach for long pose, that is lack of reported cases involving a dual approach, that is patient specific implant for long bones and porous TPMS implementation. So when long bones are treated by porous implant, there is two issues which can be generated, like mechanical requirement which cannot be achieved by completely porous long implants. In that case, we have introduced this TPMS to achieve the unique mechanical properties which can also be used for long bones. Virtual post-operative systemic approach, that can also be achieved by we have proposed one semantic algorithm by which we can do this type of post pre-operative systemic approach for developing patient specific implants. So by identification, like by study of TPMS, we have also found their pore sizes is also in the range which is suitable for the tissue in growth, that is 300 to 600 micrometer. Lattice dimension that can also be achieved, that is greater than 10, so that is also beneficial. Porosity range then that you can achieve from 10% to 90%, or whatever you want according to your need. So this can also be additively manufactured. So due to the advent or due to the discovery of additive manufacturing technology, these type TPMS based implant is only actualized. Before that, that's why TPMS was cannot be manufactured or developed. So that can also be manufactured by several electron beam melting, selective laser melting. So elastic modulus rays can it can also be achieved in the cortical bone range, that maybe 4 to 30 GPa or 0 to 0.2 to 2 GPa for trabecular bone. Yield strength can also be achieved for cortical and trabecular bone. Was algorithm which are used for developing several image-based uh, image-based porous structure, that can also be used, that is marching cube algorithm. That are the advantage of using these type of TPMS. And permeability you can also achieve in the range of which is required for cell proliferation, that is zero point 0.5 into 10 power minus 9 to 3 into 10 power minus 9. And it can also be treated for it can also be used for treating the large segmental defect that is greater than 3 centimeter. And you can also use the any type of metals which are used for biomedical application. So these are the unique characteristic of TPMS. So in this work, we will present the conversion of STL to solid of TPMS, stress shielding, how we have reduced. I will show integrated geometric and biological properties like morphological properties, fluidic properties, and mechanical properties. We will show, and how we have developed the novel. We have done a case study. Like in that, we will show how we have developed a patient-specific implant on demand, and how we have mapped the patient specific implant on to reduce the weight of any solid implant. So this is the visual summary of this presentation. Like this is the type of porous structure based on the TPMS. Like we have involved this four: primitive, gyroid, diamond, and IWP. See, these are having the unique characteristic. So mechanical analysis was done by putting the compressive load, and fluidic behavior analysis was also performed. Accordingly, we have shown the reduction of stresses. We have shown the increase of stresses on the surrounding bone where porous TPMS based porous implant was used, and finally we have developed one porous based implant for treating the segmental bone defect. So how we can do this mathematical modeling of TPMS based porous structure? So there are several type of mathematical modeling which are mathematical modeling approach that are used to develop certain type of structure. But in this case, we have uniquely modified the two combined the two approaches. Like this is the analytical method that in that you can use the parametric modeling and implicit modeling both. And when you are going for finite element based method of modeling, that is image based modeling, CAD based modeling, and homogenization theory. So based upon the strategy what we have involved in this study, we have merged both the methods, like analytical method and finite element based method. That is we have used implicit modeling and CAD based modeling both to develop one TPMS structure. So, what in implicit modeling we have used several set of trigonometric functions of particular TPMS. Like for primitive, we have used this cos x, cos y, and cos j. Accordingly, there is set of trigonometric functions which can be used to develop these type of structures. So in that sense, it is not possible to develop these type of structures into the CAD model. But we have used the CAD tools to convert this model into the solid model. Since we are using several MATLAB subroutines to run these equations, and finally we are developing the STL and ISO surfaces. These ISO surfaces are uniquely converted into the models, that is in the form of STL models. So that model can only be used for printing. That STL model we have used this CAD based modeling technique to convert into the solid model. So in this equation there is a constant C that controls the volume fraction of each TPMS structure. So there is very important role of this C. You by slightly varying the values of C, you can change the volume fractions, you can change the pore sizes, porosity, or any thickness of the strut of the porous structure. Anything you can achieve. So this is the representation where we have shown like if you are lowering the value from -3, or -3 or +3, that will lose its interconnectivity. Means if you'll see, these type of structures is connected with each other, and if you are varying the value of C by in particular case of IWP, like from -3 to more than 3, so it will lose its connectivity. You cannot connect the structures what you have developed. So but accordingly, you have to keep this C value between -3 to 3, and you will achieve a very fine and unique volume fraction according to your specific need. So later on, like by the using of subroutines, bilayer surface generation can be done, and later on it can be capped. Uh, it can be capped. Both surface can be capped to get a complete TPMS model. So this is the triangulated closed implicit surfaces. For example, this is the gyrite case, and I am just showing for the gyrite case. It can be applicable for all the structures, this where whatever it is shown for diamond, primitive, IWP, gyrite, and there are many more also. But previous study have found like these four structures only show the unique characteristic for application into the bone. So that's why this work only adopt these four structures only. Next, when you are getting any STL model, that needs to be optimized for converting into the solid. So there are several set of error that can be that are achieved during the running of MATLAB subroutines. So, like missing triangle, you can see disjoint body can also be generated. Connected triangles can also be generated. Inverted triangles can also be generated. So there are many set of errors which can be generated. These requires a set of optimization. So there are several techniques for which we have given one algorithm to. So by which you can optimize these errors which is generated after running the MATLAB subroutine. So this is the optimized STL model which was achieved for Gyrite. This is the subroutine which we have. This is the algorithm which we have followed for converting STL model into the solid model. Like in previous slide, we have shown the capping of the surface. This is the optimization of triangle or the facet triangles. That is again converted into the nerve patching. That is nerve, that is non-uniform rational B splines. Again, you are fitting the STL to estimate what changes. Is there any change or whatever changes that has been done after the solid conversion. So there in this sense you can see there is very slight changes. There is certainly there will be slight changes, but it happens that fraction is very low. If you are talking about the 80% porous unit cell, so there will be plus minus 2% of variation. Okay. National so now after that you have to cap this surface, and finally you will get this model. So this is the total subroutines what we have followed to achieve this solid model from the STL. So these are the corresponding converted as solid models. For example, this is for unit cells for gyrite, this is for P, and this is for diamond, and this is for IWP. So unit cells we have adopted 2 cross 2 cross 2. That is one that that is not unit cell. Sorry, unit cell what we have adopted, that is 1 cross 1 millimeter, and the structure we have used for analysis, that is of 2 cross 2 cross 2. So that is aligned with some ISO standard. Uh, so further morphological, mechanical, and fluidic response of TPMS structures, which was achieved, that was also done. So, what are the effect of morphological parameters on biological properties? That also, if you'll talk about the porosity, it affects the cell migration and proliferation. Wall thickness, if you'll talk about the thickness of any porous structure, that also affect the biodegradation rate. If you'll talk about the surface area, what should be the surface area and what is the impact on the biological property? So it literally affect the cell growth and increases the biodegradation rate also. And if you talk about the porous shape and sizes, that also affect the uh, cell attachment, proliferation properties, etc. So there are many more which can also be accounted. That depends upon the morphological properties. So there are set of morphological measurement for pore sizes. Like we have measured by this, we have fitted one ball on the pores of each primitive garrite diamond IWP, and the length, the dia of that ball we have measured, because that having some uneven surface dia. So we have fitted this. Ball on these pores. So what is the best fitted? We have calculated that diameter and thickness that is represented. We have measured the thickness by this. Morphological study and mechanical and fluidic study, we have incorporated volume fraction, that 10%, 20%, 30%, 40%, and 50%. So if you'll talk about the volume fractions, when volume fractions is uh, increasing, porosity will be decreasing, or either that is vice versa only. So that type of choice what we have made for adopting these volume fraction only, because earlier studies have shown in this volume fraction only you can achieve the appropriate biomechanical parameters. Later on, morphological studies was also performed on total of 20 structures was used for this study, and 5 set of each unit cells we have adopted. So these are the set of morphological properties which are achieved for PGD and IWP. If you talk about the porosity, that is ranging from 40, 50, 60, 70, 80, 90, and that also having the pore sizes that is ranges from 440 micrometer to 330 micrometer. So greater the pore sizes, that is more beneficial for bone integration into the porous type of implant. So if you talk about the 40% porosity, that pore sizes is very low. So in that sense, we have analyze all the models, and according to the fluidic parameter analysis, according to the mechanical parameters, we have adopted a best choice of porous structure for biomedical application. So if we'll talk about the pore sizes, that will be when porosity will be increasing, that it will be also increasing. And if you'll talk about if you'll compare between these four porous structures, so P structure having the highest pore sizes among all porosity level. If you'll talk about the 40% porosity, it is still having 250 micrometer of pore sizes. And if you'll talk the highest porosity level, that is 90%, that you will see that having the highest round about 430 micrometer. And I Gyrite experienced the lowest pore sizes, but still, if you compare about the gyrite and other porous structure, gyrite having some other unique properties that cannot be replaced by the other TPMS based porous structure like PD and IWP. In that sense, Garide having some other advantage, just for example, its unique interconnectivity. Its unique in interconnectivity of the model that also gives rise for the cell attachment and cell proliferation properties. So if we talk about the surface to volume ratio, so achieving more surface area when we are applying any material on the body, that is more beneficial. But at the same time, if you are getting more exposure on the surface, so it can degrade in a more better way. But if you are taking a non-degradable material for application into the bone, in that sense, larger surface area sometimes that is not beneficial also. But if you will talk about this porous structure, uh, that D experience the highest surface to volume ratio, but and P experience the lowest. And if you'll talk about the comparison between the thickness and surface area, that if you're talking about the thickness, if you are increasing the thickness, that surface area to volume ratio, that is as YVP, that will always be in the decreasing mode. If you'll talk about the compare the thickness with porosity, that is having the same trend. If you are increasing the porosity, like thickness will be reducing accordingly. You have to tune the any type of implant which you are adopting. So these are the important morphological properties which we have to compulsory account before designing any type of implant. The same thing, what these are the concluding remarks what we have discussed. Mechanical performance in evaluation of TPMS lattices. So we have done one analysis for all four structures for different porosity, and we have adopted this materials what we have taken into the study, that is titanium Ti6L4B based material that having the elastic modulus of 104 GPa, and that also having that property is adopted from the additively manufactured titanium alloy. So same we can also use during the experimental study. So yield strength, that is 1098 MPa, and density that is having 4.40 kg per meter cube. And we have done the simulation according to the ISO standard, that is 13314, that is mechanical testing of metals, ductility testing, compression test for porous and cellular metals. So in that sense, we have made the unit cells for the study, that having the 6 times more than the unit cell size. So for that we have taken a total of 8 unit cells, of size of 2 cross 2 cross 2. And again, when meshing was performed, before meshing, like we have done the mesh sensitivity analysis. In that we have taken zero point 0.1 millimeter mesh sizes, 0.07 millimeter sizes, 0.04 millimeter sizes, 0.02 millimeter sizes. So finally, we have adopted the universal size of 0.04, that reduces, like we have to take account the both things. You have to consider the solution accuracy also, and your computation limit also. So according to our computation limitation, we have adopted this 0.04 millimeter of size, that gives a very outstanding results. And solid 186 tetrahedral mesh are used to mesh this model. So loading condition, we had adopted same loading condition that was given by this ISO standard. Same we have adopted. That compressive axial displacement load was 0.1 millimeter. That was kept by putting a force plate top and bottom area, and it was sandwich the model which we have analyzed, all four model which is sandwiched into this plate, and the loading was done. The model was loaded upon top. So what are the results which we have obtained? There are many unique results which we have obtained. So for this, we have only shown one result, one result for all the models. So if you will talk about diamond, garide, IWP, and primitive structure, a maximum force of 194 Newton is required for D, and for G that is 185 Newton, for IWP that is 183.44, and for P that is 123.8. So in that sense, you can say that D is the strongest material for particular porosity. So these are the contours which are also observed during the deformation of these materials. So if you will talk about the at particular load, what was the deformation at particular porosity? Just for we have shown if for a 70% porosity, if we'll talk about the equivalent strain at 419 Newton, this is the values of strains which was observed. So if we'll talk about that is very low into the gyrite, and we have calculated this effective uh, elastic modulus. What was the effective elastic modulus? That was actually came after introducing the TPMS porous structure into the solid space. So if you'll talk about the 50, 60, 70, 80, 90, so literally if you'll go about the increasing of porosity, the elastic modulus has been reduced. So if you talk about IWP, you can see the trend that is always in reducing trend, that is 34, 23, 14, 7, 2. And if you talk about the primitive, so there is net reduction among all the TPMS structures. For you will talk about the primitive structure, that is the highest reduction in the young modulus. So if you will talk about the application sense, this P structure can also be used in the replacement of spongy bones. And if you will talk about the structures which can be used for cortical bone, that G structure can be used. Or if you will talk about the net reduction, overall the trend was very high. The range which I lie for a cortical bone, that is 4 to 40 JPL. So every material can be applied for replacement of large bone segmental defect. So yield compressive strain that having the same trends according to the Young modulus only. So that having the decreasing trends when porosity is increasing. Energy of absorption properties, that is also very unique for primitive and gyrite. If we'll talk about the energy absorption criteria, that for primitive, that is also very outstanding, and for gyrite it was slightly lower than primitive, but still primitive and gyrite both can be used for medical applications or scaffold or manufacturing of implant, because these type of reduction are uniquely attributed to these type of absorption properties that are uniquely attributed to the resilience of any type of implant. So it can absorb the energy during the physiological loading. So these are the same conclusion what I told. And now we will come with the fluidic behavior analysis of TPMS structure. So for fluidic behavior analysis, the fluid domain was also generated along with the fluid scaffold. And this is the Garide scaffold which was used into the mechanical analysis. Same, it was adopted into the fluidic analysis, and domain was also generated by Boolean operations. And later on, this is the this is the final model which we achieve for analysis. Same, it is also represented for the Garrett geometry, and same we have taken for IWP and diamond. So this is the complete model setup of the fluidic geometry. These are the number of elements which was generated. Like to evaluate the fluidic properties, we have only adopted the water. Right? This is the property of water, that is density, that is 1000 kg per meters per, viscosity, that is 0.001 Pascal second, that is mesh size, that is same we have adopted what we have used into the mechanical analysis, that is 0.04 millimeter. So there are unique properties which was obtained during this analysis. So I will directly go to the results. These are this porosity. When you will talk about the porosity, when it will be increasing, then you will get the higher permeability. If you will compare from 50 to 90%, if you are increasing the porosity, that higher permeability will be achieved. So wall shear stress distribution, that was very unique for each and every model. So what are the conclusions which was made from the fluidic analysis? That is, uh, no optimal wall shear stress magnitude has been presented in the literature. So the magnitude what we achieved during the fluidic analysis of these Garide and primitive, that was very, very unique. And the differentiation of osteoblast and osteocytes from mesenchymal stem cell in bioreactor system, that is a range of smaller than 50 megapascal of wall shear stresses, is often recommended. That what we have achieved into that in this type of structures. So next, that is the representation of TPMS porous structure or stress shielding. What is the uh, how we have uniquely mapped this porous structure on implant, and how we have achieved, or how we have reduced the stress shielding phenomena, that is demonstrated in this slide. So this is the schematic representation how we have performed these mapping and analysis. So patient CT data was adopted from the recruited for many patients. Like we have done the semi-automatic. We have used the semi-automatic algorithm to develop these bone models. Like this is the representation how we have done the image processing, and we have done the calculation of 3D model. And if 3D model is achieved, that is having so many errors that needs to be optimized before using these model as FEA. So STL model export was done, and we have remeshed into the 3-matic software, and we have removed many error of the triangle, like facets, holes, overlapping triangles. There are several errors which are removed. And finally, this is the STL model, which is converted into the solid model. So this is the cross section view that you can see the uh, mesh which is generated into the volume. So this is the complete evaluation process of getting the stress shielding phenomena. Firstly, that model which we have developed, that was Fumer, that was converted into the CAD model. We have intensely generated a defect, that representative segmental bone defect is hitting 50% of the bone circumference. So earlier in starting, I have discussed that having 2 to 3 times the dia of bone, that is critical size defect. So same we have created intensely, that is almost 50 millimeter size defect. For that we have this we have extracted from the model, and we have taken a large size lattice of primitive, and we have booleanly operated, and this is the final model that is uniquely matched with this anatomical location of the bone. So this is the final model which we have achieved to actualize the stress shielding phenomena as well as to actualize the TPMS application on the bone. So this is the same representation, like bone resection have done, unit cell was adopted, we have made the lattice structure, segmental defect was taken from the bone intentionally, and it was Boolean intersection was performed, and we have achieved this mm, uh, porous scaffold. So after analysis, like this is the complete setup set model which we have adopted for analysis. Like material properties we have adopted, that is orthotropic property of the bone we have adopted from the literature. Like element size for the femur we have taken 2 millimeter, for scaffold we have taken 0.3 millimeter, and solid scaffold that we have taken for 2 millimeter size element size. And total mechanical material properties which we have adopted for um, titanium alloy, that is same which we have adopted for the earlier study, that is 107 GPa of elastic modulus, and properties that is of both. So loading was done in a unique fashion, like lower condylar region was fixed, and one hip reaction force was applied, that value is given into the XYZ coordinate, and hip muscle forces were also applied at greater trochanter. So this is the same close view of anatomically matched scaffold. So this is the results which we have found. Like if you'll talk about the solid scaffold, this is the solid scaffold representation, and this is the porous scaffold representation. So during the use of solid scaffold, the stresses which are transferring from the adjacent bone, that is very low. And if you are adopted the porous scaffold, like in this case, in this study, we have adopted the uh, porous scaffold that is primitive. So this is very much increased. That is, if you are comparing with the solid implant, that is 76. If you are comparing with the porous implant, that is 325. So if you are matching this stress transfer on the bone, that is very high. So by this stress transfer, we can estimate there will be certainly a reduction into the stress shielding phenomena after using this type of primitive implants. So this is the same thing. So next, finally, we have developed a conceptual patient-specific model to treat the tumor femur bone. That was actually a patient conditions which we have adopted for this study, and it was ethically approved from the our institution ethical committee. So this is the subject bone that having the actual femur condition, that is almost equal to the large that lie into the large bone segmental defect cases. So if you compare with the sizes of bone defect, like if this is the tumorized bone, like resection of bone by the surgeons are done far from the femur, because if you are taking such type of mm, implants closer to the femur, tumor, that there is high chance of getting the femur tumor infection again into the healthy bone. So for precaution and for safety, for superior outcome, the more bones are laterally removed. So accordingly, we have computationally removed the patient's tumorized sections, and we have also removed. Um, we have taken the one plane. Both plane are that are used to cut the tumorized bone, and we also try to preserve the femur shaft. that is femur shaft in conventional surgery, that is all removed. So that is reconstructed by some other implantation method. So this is the patient specific fitting of that conceptualized model. So this is after that we have extracted the final hip implant large bone segment model. So same thing like surgical planning of bone tumor resection. It was done in a very precise way. Like we have done several cuts to get removal of this tumor bone, and the porous based implant should be fitted in that removed section. So this is the conceptual model of patient-specific implant to treat this type of cases. So this is the prototype of model what we have made, and we have again we have mapped into the same patient which is having the femur, and it was uniquely removed. Whatever extra material we have removed from the implant, and we have get this final model. So this final model that is uniquely matching the patient anatomy. So there will be no other complications when it will be applied into the body in respect of mechanical responses, and to protect the neurovascular structure, to protect the muscles which is surrounding with the these bones. That is that issue can be resolved. So this is the final prototype, that is distal condylar. In this distal condylar, we have made two sections that can be easily inserted also. This implant can be easily inserted, and that can be easily removed also. So proximal bone shaft in area, that we have taken one part, and distal condylar region we have taken second part. So this is the final model of both the parts, and we have uniquely mapped the primitive surface on this uh, um, design, conceptual design of patient specific implant. And like this is the prototype model. If we are trying to get this study in some other animal models, so this is the final model that having the, we have also used studied several other parts like biological studies is also done in this. So we have achieved several type of good responses. So finally, this conceptual model can have the capability to filled by the bone graft. There are several advantages that can be achieved by this type of TPMS based porous implant to treat the large bone segmental defect. So software that we have used, that are many, that is Materialized Mimics, Materialized Schematic we have used, ANSYS we have used, ANSYS Space Clamp, Workbench, MATLAB we have used, Creo 3.0 that is used, FreeCAD that is open source, that is also used to model the surfaces, Origin that is used to plot the uh, maps. These are my references. Thank you.
Thank you so much, sir. So dear participant, if you have any query, you can unmute yourself and ask your query to our esteemed speaker. You can also write in the chat box.
Yes, if any confusion is there, please write. So
Do you have any
this is my
query?
email ID also.
I think there is no query, sir. And but
Okay.
some participant has written, provided their feedback, like very interactive session. Not interactive, so you just make it interactive. Uh, that should be
Thank you.
informative. Muhammad Raja sir, if you have any query, you can unmute yourself.
So this is my email ID. If anyone have query or related with this presentation, I can give answer.
Okay, sir. So I think there is no query, sir. So
Okay.
again, I would like to thank you, sir, for accepting
Thank you
our
very much.
invitation and delivering such a wonderful, informative session. Talk on your expertise. So you have contributed a lot in the biomedical engineering field.
Thank you very much.
So kindly accept
Thank
our gratitude as a token of memento for this virtual session. So kindly accept
thank
it,
you.
sir.
Thank you, sir. Thank you very much. Thank you all for listening me. And if is there any issue or kind of with this presentation, I will be happy to answer.
Thank you, sir.
Thank you very much, sir.
Yes, sir. Our next speaker has also joined with us. Let me introduce first Dr. Santosh, sir. Okay.
Yes.
Ji, sir. Good
Hello.
afternoon, sir.
Good afternoon.
So it is our privilege to invite our invited speaker, Dr. Santosh Kumar, sir. Dr. Kumar is currently working as Associate Professor in the Department of Chemistry, HBTU Kanpur. He also served as a research professor in the Department of Organic and Nanosystem Engineering, Division of Chemical Engineering, Konkuk University, Seoul, South Korea. Dr. Kumar received his BSc and MSc in chemistry and DPhil in chemistry from the University of Allahabad. He worked as a research associate at the Department of Chemistry, MNNIT Allahabad. He was awarded CAS TWAS postdoctoral fellowship in 2008 and worked in Chang, Changun Institute of Applied Chemistry, Chinese Academy of Science, China. Later, he joined as a KU Brain Pool Professor and Researcher at the KU, Seoul. He was awarded FCT Postdoctoral Fellowship in 2012 and worked in the University of Coimbra, Portugal. During his career, Dr. Kumar has published more than 105 research papers in reputed journals indexed in SCI, edited 3 books, 10 book chapters, and attended and participated in various international and national events, including workshops and conferences. He also served as a member of different organizing committee and an editorial board member and reviewer at a number of the scientific journal that is indexed in SCI and Scopus. Beside, he is associate editor of Asian Chitin Journal and editor-in-chief of the Journal of Biological Engineering Research and Review. Current research interest involves the source appointment of the chemical modification, optical and biological property of chitogen biopolymer for the biomedical application, hydrogel, aerogel, nanomaterial, drug and gene delivery, food packaging, and environmental chemistry. So we should we are highly privileged that sir has joined with us in this 5 days international faculty development program. So we are inviting you, sir, to deliver the keynote talk. Over to you, sir.
Yeah. Good afternoon, everyone.
Good afternoon, sir.
So slide is showing.
Yes, sir. Yes, sir. It is moving.
Right, moving.
Yes, sir.
Okay. So first of all, I would like to thank Dr. Gaura sir, who is invited me for FDP in FDP program as a invited talk. So today my talk is Chitosan bionanomaterial for biomedical application. So this is the picture. So everyone knows MNIT, and I worked here in Changchun Institute of Applied Chemistry, China, Chinese Academy of Sciences, China, 1 year, and 4 year in University of Coimbra, Portugal, Europe. This is the very oldest university, founded in 1290. Around 25,000 individuals study in this spectacular UNESCO World Heritage Site today. So these are the view of Coimbra city. This is the
Sorry, but I think he's not audible.
so are you hearing me?
Yes, sir. Yes, sir. Yes, sir.
So chitin is the second most abundant biopolymer found in nature after cellulose. It is non-soluble but biodegradable. So this term chitins comes from a Greek word. It's called envelope. So if you can see in chitin in nature, we can find in algae, fungi, protozoa, coelenterate, bryozoa, mollusca, annelid, arthropoda, brachiopoda, phoronida, pogonophora, bacteria like rhizobia, invertebrate homologous to node factors. So uh, here and the crustacean cell. So if you see here location of chitin in Artemisia cyst, this is chitin containing as a envelope. Uh, and if you see in a crustacean cell, here epicuticle, exocuticle, endocuticle, this is the part, and here if we can see in this part, so chitin is formed in fibrillin. So this is this diameter is 2 angstrom. So polymer structure. So this is the chitin polymer chemical structure. So here you can see the dotted line indicated hydrogen bonding with the chitin poly N acetyl glucosamine polymer, and dashed line. This dashed line indicated hydrogen bonding between polymers to form alpha, beta, gamma chitin. So what is the alpha, beta, gamma chitin? If you see polymorphic form of chitin, so alpha chitin, this arrangement is one line in parallel, just and if you see beta chitin, so beta chitin have one parallel and one is anti-parallel arrangement. So and in gamma chitin you can see one parallel, so two anti-parallel, three anti-parallel, so one parallel, this types arrangement. So alpha chitin we can get from shrimp and crabs, and beta chitin squid pens, and gamma chitin we can get from fungi and yeast cell wall. So this is the structure of chitin. So here you can see NHC double bond OCH3 acetylation group. And how we can prepare a chitosan. So first we can dry this shrimp cell and crab cell, anyone. So and acid treatment to remove calcium carbonate. So calcium carbonate and in also sodium hydroxide treatment. So we can remove protein other. So we can get the chitin, and after chitin we can deacetylation with sodium hydroxide or enzyme treatment. So we can get chitosan. So here you can see in carbon 2 position NHCOCH3. This is the acetyl group, and here after deacetylation we can get NH2 group. So this is the structure of chitosan. So
डाला था वो अब नहीं देख सकते लोग
in a carbon 2
देखना हो तो
position you
कैसे
can see NH2 group.
स्क्रीनशॉट लेके
This is
भेज
the 3D model of chitosan. So similarity and dissimilarity in cellulose, chitin and chitosan. So if you can see in cellulose in carbon 2 position OH group, uh, 3 OH, here 5 at carbon 5, 5 in 6 OH group, and this is the one for beta glycosidic linkage. So if you see in cellulose, chitin and chitosan, we can see one for beta glycosidic linkage, and in this is the similarity in cellulose, chitin, chitosan. If you can see in carbon 2 position in chitosan NH2 group, in chitosan in chitin NHCOCH3 group, and in cellulose in carbon 2 position OH group. But you can see in carbon 6 position OH group in cellulose, chitin and chitosan. So this is the similarity. So properties of chitosan. So physicochemical properties, chitosan have cationic polyamine on protonation adheres to negatively charged surfaces, biomucoadhesive, and form gel with polyanions. It forms salt with organic and inorganic acid. It is compatible and miscible with other polymer like cellulose, PBA, gelatin. So in high molecular weight, it form linear polyelectrolyte. It shows chelate forming ability with certain transitional metals. It is amenable to chemical modification because in carbon 2 position and carbon 6 position there are one reactive amino and hydroxyl group. So it is a very reactive group and carbon 2 position amino group, and it is easily formed into fiber, and it has high charge density at pH less than 6.5. So in biological property, chitosan is a biocompatible, biodegradable, safe and non-toxic. It is bind to mammalian and microbial cell aggressively, and fungi static, spermicidal, anti-tumor activity. It accelerates bone formation, hemocompatible, antibacterial and antifungal activity, a regenerative effect on connective gum tissue properties. So market of chitosan product, if you see here in the chart, so 50% in nutraceuticals, so and general food 10%, and cosmetics 5%, and this is the agriculture 5%, In medical devices, very less, and feed uh, uh, feed industries 2%, flocculation 17%, and in textile industry 3%, pulp and paper 2%, and others are 7%. So chitogen, pure chitogen, some it can also used as a fat bounder, and you can see there is a many product in market. So application of chitogen. So in food industries and in food industry, we can use as a food preservation to increase shelf life of any food material, and water purification. It's also can use in dietary supplement. So like chitogen can use as a fat binder. So some country they are using to make a make a thin. And in material science, also used as a biosensor in bioimaging and uh, cosmetic industries and textile finishing, polymeric membrane. So because chitogen can modify and also it can make a nanofiber, and in textile finishing it can use as a binder um, with dyes. In medical sciences, chitogen have a very good application in hemostasis and control drug release, wound healing, wound dressing, anti-tumor, anti-ulcer application. So in microbiological, it has antibacterial, antifungal, and some many application of chitogen. In immunological, chitogen can use as a biological response modifier, gene therapeutics, polymeric scaffold for cell culture, and in agriculture can use as a soil enrichment, increased crop production, degradation product, and polymeric nanoparticle, synthetic polymer blend can also use. And this is the our work, gel and film for biomedical application, preparation, nanoparticle synthesis, physicochemical characterization, antimicrobial activity, gene delivery, and drug delivery, wound healing, and food packaging. So gene delivery or gene therapy is the treatment of human disorder by the introduction of genetic material to a specific target cell of a patient. So I will show you. So we have synthesized a chitogen derivative gel from solvent exchange method, which can reduce toxicity and enhance the biocompatibility of the zero gel aerogel. So there is a many method of preparing chitogen nanomaterial and ionic gelation, because in ionic gelation, in which in this method which oppositely charged macromolecule are conjugated for synthesis of chitogen nanoparticle, because chitogen solution, chitogen can soluble in 1%, 2% acetic acid. So that chitogen have a positive charge, and TPP, sodium tripolyphosphate, has a negatively charged. So that can here you can see this is the mechanical stirrer. So we can get ionic gelation chitogen nanoparticle with ionic gelation method. So emulsion and cross linking method. So in this tube, chitogen solution used as a aqueous solution, means 1%, 2% acetic acid, and is emulsified in IL. There is a in IL phase, and water IL emulsion, and also for stabilize this droplet, we can use a cross-linking agent such as glutaraldehyde and formaldehyde. So emulsion droplet coalescence also is a one method to synthesis chitogen nanoparticle. So here a chitogen emulsion, a aqueous solution of chitogen and drug in liquid paraffin IL, and sodium hydroxide emulsion. So we can just add this with a high speed stirrer, mechanical stirrer. Then it can be the droplet of each emulsion collide at random and coalesces, and after that centrifuge, washing, and separation of chitogen nanoparticle. So reverse missile method, we can synthesize chitogen nanoparticle using reverse missile method. So here this is the chitogen and drug and cross-linking agent. So just add with a mechanical, there is a one mechanical stirrer and surfactant dissolved in any organic solvent. So surfactant can make a nanoparticle with a chitogen solution and evaporate of solvent, then purify and dry and remove the surfactant, so we can get a chitogen nanoparticle. So this we have already synthesized a chitogen chromium derivative gel. So this is the XRD structure of pure, and this one is the pure chitogen, and this one is the chitogen chromone and derivative gel. So here we can see there is some changes. So material chitogen chromone derivative is a highly amorphous structure. So and if you see FSM images, so here we can see porous and chain like shape and cross section of randomly oriented grains. So this is the very porous structure. And also we synthesize chitogen terephthaldehyde derivative gel, and this material is UV visible, have in UV region 200, and but PL spectra of chitogen terephthaldehyde we can see 500 emission is 500 nanometer. So this material can use as a biosensor, and we also synthesize chitogen biopolymer sheaf base with crotonaldehyde. So we synthesize, characterize, and optical property reported, and antibacterial activity. This work is already published. These are the many some other work we have reported: chitogen methoxycinnamaldehyde, and we also use some pyridine aldehyde and chloroquinoline derivative for biomedical application, and also 2-thiophene carboxyldehyde. We used N-heterocyclic we prepared a N-heterocyclic chitogen derivative gel for biomedical application, and a chitogen phenothiazine derivative by microwave assisted synthesis. So that can material can use as a biosensor in sensing compound. This also we synthesize chitogen based dye containing naphthalmide group. So this this is the physicochemical property of chitogen and a derivative chitogen biopolymer dye. So this is the TGA, and this one is XRD. This is the same image. So same image, we can see that material is a porous material, and also this material chitogen biopolymer dye is the solubility test. And also if you see there is a this one is UV spectra, and this one is the photoluminescence spectra. This one is PL spectra of chitogen, and this one is the chitogen biopolymer dye at excitation wavelength 325 nanometer. And so here you can see this is the rate shifting at this emission is more than 450, and one is 550 nanometer. This work already published in carbohydrate polymer. And also we synthesize chitogen azo-based amino methane sulfonate complex. So this gel can form by a complex with electrostatic interaction between ammonium ion of chitogen and a sulfonate group on methane sulfonate. So this has also a good activity, antimicrobial activity, that compound. And this is the XRD data. So this is the for pure chitogen, and this one is the derivative. So this material is we can say this is the crystalline structure, and this is the same image. And this is the TGA. TGA is also showing its compound is thermal stable, and this one is PL spectra of chitogen azo-based amino methane sulfonate complex. So in if we can see in derivatives, so it's 450 lambda maximum. And also also we synthesize chitogen containing azo-based sheaf bases. So first this organic compound we synthesize and be conjugated with chitogen. So and in this work we have reported a thermal, antibacterial and birefringence property for biooptical devices. And also and this the antimicrobial activity. We used E. coli and Streptococcus aureus. This already published in RSC Advances. So and some other we synthesize chitogen containing azo-based material like chitogen biopolymer, and this chitogen containing azo-based sheaf base effect on optical property. And also we synthesize this preparation of diisocyanate modified chitogen. So chitogen and this the MDI, there is a isocyanate, two isocyanate at room temperature. We have modified chitogen. So here a structure is confirmed by FTIR. So here you can see this the CH2 stretching and NCO stretching, but in main compound we cannot see NCO stretching here. And this is the photoluminescence spectra, a spectrum of synthesized compound at the excitation wavelength 382 nanometer and emission wavelength 475 nanometer. So and that in this DIMC compound is a very good antimicrobial activity. We used bacteria against E. coli and Staphylococcus pyogenes. And also we, uh, we can modify bionanocomposites like chitosan, gelatin, silver loaded zinc oxide. So first we uh, used chitosan in 2% weight by volume glacial acetic acid, and gelatin in distilled water. So and mix with chitosan gelatin, and also synthesize nanoparticle nanoparticle by green method. So first we synthesize zinc oxide, and after we loaded silver, and again we modified, we just blended chitosan gelatin and silver loaded zinc oxide nanoparticle, and we poured on plate and dried, and that material uh, mm, composite that bionano composite material showed very high mechanical properties and antimicrobial property also. And we also modified chitosan and zeolite. Why we use zeolite? Because zeolite is a very cost effective material, and naturally uh, is aluminosilicate microporous mineral, commonly used as a variety of application in different area like purification, separation, adsorbent, and drug delivery, and it wide application due to the specific chemical composition and unique porous structure. And also if you change this material with other biomaterial, so it can show very good thermal stability. So and this is the preparation of chart, chitosan and zeolite mixing with and solvent exchange with acetone. So and after that lyophilize at 24 hour at, uh, for 24 hour. So we can get this zero gel, and after calcination at 450 degrees centigrade. So we can see this the composite material, chitosan and zeolite composite. So structure was confirmed by FTIR and XRD, and this is the thermal property. If you see this C1 is the chitosan zeolite. So chitosan zeolite material have a good thermal property comparison than TGA of chitosan and TGA of zeolite. This is the same images of SEM images of pure zeolite, AV, this one, and this is the chitosan, uh, and this one E and F is the chitosan zeolite composite. So this is the HR10 image of pure. This one is the pure zeolite, and this is the chitosan zeolite material. So this from HR10 we can see that compound is porous structure, and there is a pore structure here. And if you see zeolite have surface area 68.36 meter square per gram, and pore volume 0.103, and average pore size 6.07. And after modification, surface area decreases 22.53, uh, and pore volume is 0.068, but pore size is increases 12.09 nanometer. And we also used as a adsorption. So if you see CO2 uptake, this the pure chitosan have a negligible 0.2 millimole per gram, but in pure chitosan have 1 millimole per gram, but you can see chitogen zeolite have 1.6 millimole per gram. So that material is a very porous structure. So that material can use as a biomedical application. And this the conversion also, that material can use as a catalyst for convert epoxide to cyclic carbonate. So we synthesize this material convert 99.5% conversion with CO2. And also uh, uh, we modified with chitogen and in this TPPS like tetrakis 4-sulfonato phenyl porphyrin. That material is also this aerogel material is very good porous structure, and that have high CO2 absorption and also conversion, because surface area can increase if you modify with chitogen and porphyrin structure. And here we can see this is the TPPA, this is the chitogen, and this one is the TPPS 0.2 millimole per gram, and this one is chitogen modified with TPPS. It's about 0.9 millimole carbon dioxide per gram. And also we synthesized chitogen graphene oxide nanocomposite as a catalyst, and that composite material can also absorb CO2, and also we used as a catalyst to modify with epoxide to cyclic carbonate. And also we modified multi-walled carbon nanotube to here we modified COH group, and that COH group uh, conjugate with a chitogen with a coupling agent EDC and NHS. So we got this chitogen grafted multi-walled carbon nanotube. So that material can also used as a in biomedical application like tissue engineering and some other properties in some other applications. And chitogen material can modify organic modification by covalent bond. So this material can when if R here R can use as a hydrophobic group. If you can use as a OH group, many OH group, so hydrophobicity can increase, and stimuli responsive group, pH redox and thermo targeting group, a small molecule, nucleic acid, peptide, protein, and aptamers, and also here are we can use hyaluronic acid. So that can material use as a drug loading drug loaded and drug releasing capacity. So here and that you can see anti-cancer drug loaded, this drug loaded organically modified chitogen. It is very effective delivery of anti-cancer drug. So we also modified 2-acrylamide 2-methylpropane coupling with chitogen, and here is the work used as a that material MPS grafted chitogen used as a DNA to make a polyplex, and also we checked gene transfection, and in vitro gene transfection mechanism study. So primary amine of chitogen are responsible for its cationic nature and hence binding and protection of DNA for intracellular delivery. But transfection efficiency of chitogen-based gene transporter is severely hampered by its poor physical property, such as low water solubility and high viscosity. So in this study, in CSMP DNA polyplex, we used low molecular weight chitogen to making it water soluble for application in gene delivery. So AMP modified chitogen showed enhanced interaction with DNA and a high buffering capacity due to 2-acrylamide 2-methyl propane sulfonic acid and uh, leading to higher transfection efficiency in cancer cell line F549, HeLa, and HEP-G2 compared to pure chitogen and lipofectamine. In vivo study in valve C through intravenous injection demonstrated higher luciferase expression compared to the low molecular weight of chitogen. So this is the scheme of synthesis of CSMP gel. This material be used as be synthesized by Michael Edison reaction. This work is published in Journal Material Chemistry B in 2015. So this is the CD data of different chitogen DNA different ratio. So there we can see interaction of DNA and derivative chitogen derivative. So in pure chitogen, you can see it's not a very good interaction with DNA, but derivative have a good DNA interaction. And this is the FTIR data, and this one is UV data, and this is the TGA data of synthesized material. And this one is the physicochemical characterization of PGL3 CSMP and PGL3 chitogen complex. Here, this one, and this the particle size measurement in the absence of serum measured by DLS, and this one is the zeta potential, and this one is the particle size in presence of various serum percentage, and this one the FE10 image of CSMP and DNA CSMP and DNA CSMP complex. So here we can see here in chitogen DNA have a no very small particle. So but in CSMP DNA we can see is a small particle. It's called nanoparticle. So here in particle size also, if you see, so chitogen DNA complex have a less nanoparticle comparison than chitogen DNA complex by DLS. So here zeta potential is also very less comparison than pure chitogen. So this is also with this one is in absence of serum, and this data particle size is uh, that material and presence of various serum. So you can see chitogen derivative material DNA complex have very particle size is very less comparison than chitogen DNA polyplex. So this is the also with DNA polymer interaction in docking approach of this one is the chitogen, and this is the chitogen AMP. So by docking study also we can see there is a derivative, there is a many pose for DNA interaction. And this is the in vitro transfection of PGL3, CSMP, and PGL3 chitogen complex at various weight by weight ratio 1 to 3 5, and this is the pure PI 25K and lipofectamine. And also for this one, the cell line F549. So this one is DNA CSMP, and this one DNA chitogen. So if you see in F549, it's increasing. Cell viability is also good cell viability, a comparison than PI 25K and lipofectamine. And in HeLa cell also we can see DNA CSMP. It's also increasing in HIP G2 cell is also increasing with cell viability increasing with DNA chitogen and derivative. Comparison then also PI 25K lipofectamine. So this is the percentage transfection efficiency of CSMP, TGFP, and chitogen TGFP as measured by flow cytometry in F549 cell. This is the in vivo gene expression after IV injection in valve C mice. This one A luciferase expression after 4 days, showing this biodistribution of PGL3 CSMP complex. This one is the CSMP, and this dark line, and red line chitogen DNA, and this DNA only. So this also you can see in brain, in lung, this one in heart, spleen, kidney, liver. That material is very effective in brain, lung, heart, spleen, kidney, liver. And this is the proton sponge effect of CSMP, buffering capacity of CSMP and chitogen determined by acid-base titration method. And this one is a bifilomycin A1 study. So, and also we also one compound we synthesized triphenylamine coupled chitogen, and this you can see this the SIV base. This is also material is a very highly water soluble material. So we used as a gene delivery application. This also published in JMC in 2013. This is the schematic representation of the mechanism of cellular internalization of PGL3 and CSTCP PGL3 chitogen complex in F549 cell. Here you can see chitogen DNA, this TPSC DNA complex, and endosome formation. So material, our compound is high buffering capacity showing after releasing DNA. This is the endosmolysis and degraded TPSC. And also we synthesized water soluble tetra 4 carboxyphenyl porphyrin grafted chitogen for gene delivery application. Why we used chitogen porphyrin material? Because porphyrin have a nitrogen, so nitrogen can enhance nitrogen can interact with DNA. So this is the 1H NMR. This is the UV visual of TCPP and CSTCPP. This showing red shifting, and this is the photoluminescence. So photoluminescence have 660 nanometer, 618 lambda emission peak. So that kind material used as a photodynamic therapy and gene delivery also. So we also used cell viability. You can see the cytotoxicity of PGC DNA, PGC DNA and chitogen DNA. So that material. This one is the cell line F549. This the HeLa. This HEP-G2. This IEC 6L929 cell line. This is cytotoxicity with time. So this cell viability is also showing enhanced comparison than chitosan. This is the in vitro transfection efficiency of different cell line F549. This is the HeLa cell line is HEP-G2 IEC 6L929 in presence of this serum. This is the pure compound, and this is the chitosan, and this is the PI25K lipo NECT DNA. So this study also shows enhanced transfection efficiency, and also we modified chitosan N4-hydroxyphenyl methacrylamide derivative as a drug and gene carrier. And this the various conjugated derivative of chitosan synthesized via Aza-Michael addition. So you can see this one is the our product, and this already reported that material. So because a phenolic group, when we increase on a chitosan backbone, so it enhance its hydrophilicity, and this lead to creation of excellent loading space for encapsulation of anti-cancer drug like curcumin, doxorubicin, etc. Protection of sensitive drug from environmental stimuli, prolonging of drug circulation time, prevention of side effect of drug on normal cell. This is the formulation of chitosan derivative nanoparticle and curcumin loaded chitosan derivative nanoparticle. So just we have used this derivative and then curcumin with ionic gelation method. So we can get drug loaded chitosan derivative nanoparticle, and after that this material can use as a drug delivery application. So estimation of curcumin, we can use this equation E percentage W1 minus W0 W not and W1 into 100. So here where E percentage is a drug encapsulation efficiency, and W1 is the total amount of curcumin, and W naught is denoted non-encapsulated curcumin. So this is the plausible mechanism of curcumin loaded and chitosan nanoparticle and chitosan and derivative nanoparticle. So this is the TPP, so that can interact with pi-pi interaction, and here you can see intermolecular hydrogen bonding interaction, and this showing maximum curcumin loading efficiency of 77.03% was recorded. And this the release of drug pattern of curcumin loaded chitosan derivative in PBS at different pH value. So we used pH 5 and pH 7.4. So and this in vitro release curcumin was determined at two different pH via the direct dispersion method at a predetermined interval up to 48 hours. So in here, if you see in vitro experiment reveal that the drug release rate from chitosan derivative nanoparticle at pH was very faster than pH 7.4. This is due to the fact that in acidic environment of the chitosan derivative polymer, that matrix swell due to protonation of amine group of chitosan. So that's allowing curcumin to release out at a faster rate. That's why in at pH 5 it's showing faster than the PS7.4. So this is the cytotoxicity of polyplex DNA and chitogen NHMA complexes at various weight by weight ratio 1:1, 1:3, 1:5, 1:10. So this is the A549 cell line, HPG2 cell line, and this one. This one is the HeLa cell. So in all three cell line, increase the polymer to polyplex DNA ratio had no major effect on cell viability, implying that the chitogen CSNHMA is non-toxic even at a high concentration. If you see uh, at weight ratio 1:10. This one is the chitogen derivative DNA polyplex. So this is the in vitro transfection of polyplex DNA chitogen derivative nanoparticle and polyplex DNA CS. So this is showing this this color. So this also different weight ratio 1:1, 1:3, 1:5, 1:5. This is also showing enhanced transfection of chitogen and derivative with DNA polyplex. So mechanistic find for the enhanced transfection efficiency of CSNHMA. So we used two experiment, acid-based titration and bafilomycin A1. So in acid-based titration, so the higher H ion absorption tendency of chitogen derivative. So the amine group in the polymer provide the vector with buffering ability by absorbing H ion in the endosome and causing endosomolysis, and the effect of NHMA substitution on the proton capturing potential buffering capacity of chitogen was investigated using acid-base titration against one normal HCl. The results showed that chitogen derivative has a significantly higher H ion. Here you can see ion neutralization tendency than native chitogen. Because of NHMA, moiety is responsible for the absorption of H ion, which result in higher buffering power. The result of the titration inspired us to investigate the impact of buffering capability on transfection. And this is for bafilomycin A1. So in future perspective, chitogen-based nanoparticles are highly promising carrier for efficient drug delivery or gene delivery to tumor site. Despite the impressive progress in chitogen-based bionanomaterial, other structural and functional modification or combination with new biomaterial still remain to be explored with respect to drug or gene delivery application of chitogen in the near future, especially in clinical trial in view of safety and efficacy concern. Moreover, we believe that the widespread information gathered over recent years about chitogen along with the advancement in nanotechnology will revolutionize the design of safer and more efficient in drug delivery system for cancer therapy. It is worth mentioning that the clinical of chitogen-based drug or gene vector is still limited due to their potential risk. Therefore, development of novel chitogen-based bionanomaterial with appropriate properties such as cheaper production process, high drug loading capacity, and a specific selective delivery, controlled release of drug or gene molecule, high transfection efficacy, and finally better treatment value with trivial toxicity and undesirable reaction can significantly improve both the quality and duration of the life of cancer patient. And also you can use as a biosensor and tissue engineering and as a wound nano dressing, etc. So this is the acknowledgement of University of Coimbra, Konkuk University. So here I am working in HBTU Kanpur. So thank you.
Thank you so much, sir, uh, for such an informative talk. So you have covered up all the thing related to the chitogen synthesis, characterization, and their a huge biomedical application. So, dear
Any
participant,
question?
if you have any query, you can unmute yourself and ask your query.
Yeah, may I?
Yes, please.
Yes, sir.
Yeah. First, thank you so much, Dr. Gaurav, for such an amazing FDP because it's so wonderful to learn all these new things. And thanks to Dr. Santosh for this wonderful lecture. Actually, I just want to know that as you were mentioning about most of the studies in, will this cortisone has any immune elicited response or something like that?
Yes, if you modified with chitogen with other organic material, so it can be used as a
No,
immune.
I just yeah, because most of the things if we are just conjugating chitosan with some material,
Yes,
so is there any possibility that our body rejects that?
yes, it's a possibility.
Okay, so and the second question is that this cytotoxicity that will chitosan individually have any cytotoxicity or only when conjugated with some other things
No,
then
yeah, if you use a chitogen, pure chitogen have a, some cytotox, but if you modified with so chitogen have a no cytotox.
okay. So in that case, if there is any immune rejection from the body, so how we can go with these kind of studies, or you can say that biological activity, because there might be a chance that our body will reject this cortisone.
No, if you modify, so chitogen is very soluble in water soluble. If because chitogen is not a soluble, water soluble, so first we should modify chitogen uh, derivative with some other material.
Okay, thank you, and thank you, Dr. Gaurav, once again for
Thank you
providing
so
this
much, sir.
opportunity to learn.
Thank you. Sir, there is another question from Dr. Satyajit Tripathi, sir. He has asked that which is a better nano drug carrier, chitogen or derivative of chitogen?
Derivative of chitogen, not chitogen.
So Satyajit Tripathi, sir, you can unmute yourself and if you have further any query, you can ask. Okay, any other query? So I think there is no query, sir, still. If you have any query, we are waiting for 1 minute. Please unmute yourself and ask your query to our speaker.
Yeah, you can ask a question. I can give you my mobile number and email ID. Yes.
So I several participants Okay. Sanjay Kumar Misra sir is asking uh,
Yeah,
by adding the
any
chitogen
no,
to
yeah, any acid we can use, but 1% acetic acid is very good. Any acid can be used.
any query, sir? You can unmute yourself.
Yeah, because chitogen is soluble at pH 3.5.
Excuse me, sir. Am I audible?
Yes, yes, yes.
Sir, can we use chitosan as a nano drug delivery for the targeted tissues?
Yes, 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 but
Is there
not
any?
pure chitogen. If you
Sorry.
modify it, because chitogen, you can see chitogen is soluble at only acidic medium. So you should modify chitogen which can soluble in water, right? So
Okay.
because
And
we have modified to 3,5 chitogen derivative, which is very water soluble. But pure chitogen is not very water soluble. It can soluble at a low pH below 4, 4 or 5.
no, suppose I am having some drug, okay, and I want to
Yes,
target it a particular tissue. So I want to
yes, yes.
use chitosan as a nano particle. So can we use as a nanoparticle?
No, no, pure chitogen you cannot use, I think. If you
Okay.
modify that, yes, you can use, but main thing is solubility.
Mhm.
Any
Solubility.
other
It
query?
works perfect. It
Yes,
perfectly work at which pH?
uh, pH 5, 4, 4, 3, this pH, and 5 also 6, less than 6.
Okay. So there is no side effect while we have to
Yeah,
use.
yeah, no, no side effect.
Okay.
Any other query? Okay.
Yes,
Prushti
it
sir.
can, because when you uh, extract chitogen with various organisms, there is a changes, means molecular weight changes. So depending on molecular weight, drug delivery can also change.
Prushti sir, you can also unmute yourself to ask. Okay. So if there is, okay, yes, sir, which one is better? He is asking for, sir, which one is better source?
So,
Which one is cost effective? You can use alpha chitin. Chitin is also good, because some gamma chitin is very expensive. So low molecular weight is good. Low molecular weight of chitogen.
Okay. So again, once again, sir, thank you so much for accepting our invitation and delivering such an insightful and wonderful informative talk. And indeed, it was very interactive session also. Several participants has asked their query, and you have given a satisfactory answer to them. So now you can accept our e-memento on behalf of Galgotia University. Please accept this e-memento, sir.
Thank you very much.
Thank you. Thank you so much, sir, once again. So our next speaker has joined with us, Sonali ma'am. Yes. Am I
Hi, can
audible,
you
Sonali
hear me?
ma'am? Yes.
Yeah, you are audible.
So this is our last session, and we are highly honored and feeling very privileged to invite our speaker, Sonali Das ma'am. Dr. Sonali Das ma'am is currently working as a postdoctoral visiting fellow at National Heart, Lung and Blood Institute, National Institute of Health, Maryland, and she has also worked as a postdoctoral research associate in GCRF NTD network, that is joint collaboration of Durham, UK and India, under the Professor Nahid Ali, CSIR, Indian Institute of Chemical Biology. She also published several research papers in a highly indexed, reputed journal, highly impact factor journal, and she has received PhD in biotechnology from the Indian Institute of Chemical Biology, University of Kolkata. After that, she has um, um, received several type of skill and expertise in animal handling and patient sample handling, tissue and mammalian cell culture handling, cell and molecular biology techniques, biophysical characterization and in silico study and bioinformatics tool and techniques. So she has also published several research papers in various international and national conferences, um, and she has received several type of fellowship from the India as well as from the abroad. So we are very honored to invite you, ma'am, uh, to deliver your talk. So over to you, ma'am.
Thank you so much, Dr. Gaurav Kumar. So, can you see my slides presentation? Okay,
Yes, ma'am. Yes, ma'am. We can see.
okay. So, shall I start?
Yes, yes, please, ma'am.
Thank you so much for inviting me in this interactive and beautiful career development session. So, this is the title of my talk. Today, I'm going to talk about one of my PhD research work, which I did in India, CSIR IICB. Right now, as Dr. Kumar said, that I am a postdoctoral fellow in National Institutes of Health in USA. So, my project was about understanding the role of microRNAs in visceral leishmaniasis, which is a very well-known and well-studied endemic infectious disease, and how a particular type of macrophage polarity or plasticity is involved with this infection, as well as how what is the role of microRNAs in regulating that. So, before going into details of my work, I would like you all to get quick details about what is the disease, how it happens, and what is the causal agent or the parasite or infectious agent that causes this disease. So we all know that this visceral leishmaniasis is natively known as Kala-Azar. Why? Because it causes a black fever, and it is basically caused by the bite of a particular type of insect which we call as sandfly, which belongs to the genus known as Phlebotomus. And when it infects human by sandfly bite, the parasite enters the human blood. So the form of parasite that mostly enters from fly to human is mentioned or is denoted by image which is in left, and I mentioned the name of the form as promastigotes. So the type of image that I'm showing you all is basically Giemsa stained image. So Giemsa is a dye which stains nucleus or nuclear DNA of parasites. So whatever images that I will tell you guys in my follow-up slides, telling that it is a Giemsa stained image. So what does it mean? That we generally stain either forms of the parasite using the dye, and this is a very gold standard method of understanding parasite burden in patients as well. So in the left, you can see that the promastigote form of parasite which enters the human body when sandfly bites a human, and it is a flagellated form of the parasite, and the parasite has two nucleus. So it is kind of one nucleus is the nuclear DNA and another is the kinetoplastidial DNA. So you can see here that there are two purple dots in each cell body. So the kinetoplast is nothing but the mitochondrial representation or the mitochondrial DNA representation in parasite. Now, what happens during the life cycle? The promastigote form enters the human blood, and then from there it gets engulfed by mostly by the neutrophils from the peripheral blood. And then the neutrophil, which is containing the parasite, is engulfed or is uptaken by macrophages. Now, what is the role of the neutrophil in between? So the neutrophil basically helps the parasite to get a safe passage inside the macrophage, and the method of entering from sandfly to human macrophage via neutrophil is also known as Trojan horse model. So by this Trojan horse method, promastigote enters into the macrophage, which is the final home for the parasite. Now, inside the macrophage, the promastigote transforms into a afflagellated form, which is denoted by the Giemsa stain picture in the right panel, which we call as a amastigote. Why? Because it lacks the flagella. And here you can see, so this is a Giemsa stain of a VL infected patient or Calazer infected patient's splenic aspirate. So the bigger nucleus are splenic macrophage nucleus, big purple blotches, and the small dots which accumulated together here is the parasite nucleus, or which we call as a amastigote. So when we try to understand what is the parasite burden or parasite infection load of any infected sample, we do Giemsa stain of the sample, and then from there we generally count or calculate the number of amastigotes present in that particular field. So this inside the macrophages, you can see that the parasites can multiply like this, proliferate over there, and lyse one macrophage, and then in again enters into another macrophage, and so on so forth. Like this method, the parasite amastigote form keeps on proliferating inside the human macrophage until another sandfly bites the infected human, and from there the same cycle restarts. Like from the macrophage, amastigote again enters into the sandfly gut, and it again transforms from amastigote to promastigote stage in the midgut of the sandfly, and it again proliferates in the promastigote form in the midgut of the sandfly, and the number of the parasites which matures from the midgut then moves to the foregut, and another round of cycle starts when the sandfly bites another healthy human, or so and so forth. So this is the whole story of the parasite life cycle. So there are different kinds of this parasite which is which belongs to the genus called Leishmania. Now, different kinds of Leishmania species exist which causes different forms of Leishmaniasis. Here I will be talking about Leishmania donovani, which causes visceral Leishmaniasis. So this is the outcome of the global cases that has been recorded by WHO in 2019. So it says that around 0.5 to 1 million new global cases keeps on arriving. And the main thing, or the main thing to be why we are studying this disease is the region where we live, like I used to live, and also the northern and eastern part of India, that is West Bengal, Bihar, UP, belongs to the endemic belts for the disease. So most of the cases or most of the patient sample that I used to receive during my PhD studies, we know that cases comes in the hospitals and mostly reported from this part. So you can see the outcome of the infection, like in the right panel, there is a representation of one kid who is infected by this disease. So here you can see that there is a big belly of the kid, which is caused by bigger spleen, because the parasite mostly enters. If I just go back, so the parasite mostly enters mostly in the spleen, liver, bone marrow, and lymph nodes of human. So the outcome of the infection results in a bigger spleen, which is known as splenomegaly, and the discoloring of the skin. So the skin color starts getting more blackish. That's why it is called as scalager. And also it causes high fever and loss of weight, and also there is a huge amount of immune suppression. That means any patient who is infected with VL will be further infected by different kinds of co-infections. The most important and tragic kind of co-infection till date reported with VL is VL HIV co-infection. So there are a lot of research which is going on globally, where people are studying how to stop or prevent VL HIV co-infection, because that is far dangerous than the normal VL alone. So what my study was to understand or to find out a method where I can target host factors. So as you can see that for making the infection or the infection situation worse, macrophage has a lot to do. The parasite mostly enters and in the macrophage, and it makes the macrophage as their home. So macrophage is mostly suppressed or is basically hijacked by the parasite, and all of its system or the machinery to save the host get failed. So what is the mechanism that parasite takes over inside the macrophage? What are the macrophage molecules that parasite involves while proliferating and replicating in the human host? That was my interest during my PhD. So I tried to find out what is the role of macrophage plasticity in VL infection, and what are the important microRNAs that are involved in it, and what are the transcriptional regulation pathways which is also involved in this process. So what is microRNA? The microRNAs are very well-known and interesting type of non-coding RNAs. So beside the normal mRNAs that encodes for the protein in our body, there is a huge amount of human genome which codes for junk RNAs, or also known as non-coding RNAs. So microRNAs is one such class of non-coding RNA which is mostly transcribed initially as a prime microRNA, and from there it is spliced off and it produces a hairpin structure which we call as pre-microRNA, and from there it forms a RNA protein complex with another protein called RISC, and then this RNA protein complex can go and bind with target mRNA. So the microRNA basically aligns with the 3' untranslated region or 3' UTR of the target RNA and subjects that target RNA for degradation. And sometimes it also binds with the 5' UTR of the target RNA, or the 5' coding region of the target RNA, and it causes suppression. That is, it never allows the mRNA to get translated. So it basically blocks the translation of the target RNA, or it can cause degradation of the target RNA. So in any other means, the microRNA basically causes suppression of its group of target RNAs. So there are each microRNA can target numerous number of pathways and genes. So here I'm focusing on a particular type of pathway, which we call as macrophage plasticity. So we all know that the recent theories are coming up that depending on the microenvironment stimulus, like the type of cytokines, the type of infection that a macrophage receives, it can proliferate and differentiate into two different subtypes. So one is called as M1 macrophage, which is a classical macrophage or a pro-inflammatory macrophage that helps in activating Th1 type of response, or I would call as CD8 T cells, and that helps in clearance of any kind of infection, including parasite infections. And mostly IFN gamma, LPS, GM-CSF, these are the cytokines which can trigger macrophage macrophage profile towards M1 type. There is another type of macrophage, which is M2, which is also called as alternatively activated or non-classical macrophage. So this has anti-inflammatory type of nature. It helps in activating Tregs and CD4 T cells, which has immune suppressive functions or called as Th2 types. And this kind of suppression helps in proliferation and survival of different infectious diseases like bacterial infection, parasites, and also cancer cells. So what are the cytokines that activates M2 macrophages? Mostly IL-10, IL-4, IL-13, TGF, beta, MCSF, etc. So here I am trying to, so my first, in my first year of PhD, what I tried to do is basically to do a screen of the whole microRNA landscapes from macrophages which were infected by Leishmania donovani. So what we did, we isolated bone marrow derived macrophage from 4 to 6 weeks old biopsy mice. And these biopsy mice are a very well-known model for studying visceral leishmaniasis disease. So it can work as itself. We can infect these animals, and we can also isolate macrophage from these animals and infect those macrophages in plate. So from this bone marrow derived macrophages, we stimulated them with 20 nanogram per ml MCSF to make bone marrow derived macrophages for 7 days. And after that, we infected them with two different types of Leishmania parasites. So one type of parasite here we are mentioning as virulent or A2. What does that mean? So we generally make the parasites in the lab. So we can isolate this parasite from the infected biopsy mice, and then we transform them in a particular type of media in cell culture flask. And the freshly transformed parasite has very good activity of infecting macrophages. So we call them, and based on the number of passages that we give those parasites, so based on the how early those parasite passages are prepared, we mention the number based on that passage number. So here I mentioned it as A2, means that is the second passage of the parasite in flask. And those parasites are very much virulent in nature. They can infect macrophages with high efficiency. So we infected one set of BMDMs with this virulent second passage parasites, and we infected another set of BMDMs with 25th passage of parasites. So if you keep on passaging these parasites or Leishmania donovani promastigotes in the flask, we found that in our another experiment, published report from our lab, we found that if you keep on passaging the parasites, they will lose their efficiency to infect macrophages. So that's why we called it this A25 or the passage number 25 as the avirulent parasite. Then we purified the RNA and subjected it for small RNA sequencing. And from there, you can see that compared to uninfected or control BMGM, we got a pool of differentially regulated microRNAs from A2 and A25 infected macrophages. And from this pool, we sort out these 4 interesting candidates. Now, from there, we performed another step of RT-PCR based validation. So after doing this RNA-seq, we again infected normal BMDMs with this time with simply virulent passage parasites. We removed the avirulent passage for this time, and then we compared the expression of the particular type of these 4 candidates, because these 4 microRNAs are very well known or well studied in different kinds of immune signaling pathways in other cell types like T cells. So that's why these 4 microRNAs they caught our interest. And from there, like the RT-PCR validation, we got that microRNA 146A showed maximum fold change, like around 10-fold change compared to uninfected BMDMs. So we decided to go ahead with this candidate for our further study, and we decided to study the role of this microRNA in regulating macrophage plasticity during Leishmania donovani infection. What was known till date? So far, we know that microRNA 146A is induced by bacterial infections. And this is a prevalent microRNA which is expressed in macrophages, T cells, etc. And the main, as I mentioned earlier, that how microRNA work, it basically targets particular type of mRNA by binding to its UTR regions. So there are softwares where you can find out the target pathways of each microRNA. So the target pathway of microRNA 146A is TLR4 pathway, PPAR gamma pathway, and Notch pathway. So here I mentioned two candidates of TLR4 signaling, IRAC1-2 and TRAF6. So what a microRNA does, it basically targets these particular pathways and causes a stimulation of M1, biasness of macrophages from M1 to M2 type. So if the macrophage has higher amount of this microRNA, it has a tendency to show M2 markers or M2 profile, and which is more anti-inflammatory, and it pro-infection, pro-cancer, so like this. So we were interested to know that what happens when we infect macrophages with Leishmania donovani, and so far our RT-PCR data showed that the microRNA gets pretty high if you infect the macrophages with a parasite. So how does it happen? What are the downstream regulations and signaling that alters if you change this microRNA? Those were the part of my study. So to further understand the expression of the microRNA during infection, we performed two different types of infection model. So in one hand, we infected BMDMs for 24 hours with virulent parasites. And from there, we isolated the RNA and done the qPCR. And the other hand, we infected bulb C mice for 3 months, and 3 months post-infect, and also we isolated RNA in between. Like in my next slide, I will show what are the time points where I isolated blood from these infected mice, and from there we also performed the qRT-PCR. And after 3 months, which is the complete course of infection, we sacrificed the animal, and we isolated liver and spleen, and from there also we did the qPCR for the microRNA. So this, our main aim was to understand if we infect cells like macrophages or we infect mice, whether the microRNA is increasing or decreasing. That was the ultimate goal of our study. So in the upper left panel, you can see that the microRNA expression or the fold change of microRNA kept on increasing with multiplicity of infection, or MOI. So as I mentioned earlier, that I infected BMDMs with virulent parasites. So we infected BMDMs with different ratios of virulent parasite. So what does it MOI means? I mean, MOI represents the number of parasites per macrophage cells. So we kept the MOI from, kept on increasing the MOI from 0.5 per macrophage to 15 per macrophage. So you can see that there is a beautiful rise of microRNA expression with the multiplicity of infection in macrophages or BMDMs. And after that, the second panel represents after 3 months, we sacrificed the mice, and from there we isolated spleen, liver, and also peritoneal lavage, BMDM, and compared the expression of the macrophages in all these different kind of infection foci compared to the PBS control mice. So in the PBS control mice, we didn't introduce any infection. We just injected equal amount of PBS via tail vein route. And in infected mice, we infected like the animals with a certain counts of parasites solubilized in PBS via tail vein route. So you can see that in all the organs, there is a significant enhancement of the microRNA compared to the PBS control group. But most importantly, in the spleen, that is the second bar, that is highly enriched compared to any other infection foci, which is also quite correlating with the finding so far that the macrophage, the parasite mostly resides in human spleen and causes splenomegaly. So this data suggested that in different infection foci or different infected organs, the microRNA gets enriched. After that, as I mentioned earlier, that this is the second panel is the data of 3-month infection, post-infection. And the last panel, the third panel, is the data of different time points where we sacrifice the animal post-infection. So what we did, so 15 days, 30 days, 45 days, and 60 days post-infection, we sacrificed the animal and isolated their spleen and liver. And from there, we performed QRT-PCR to understand the expression of the microRNA. So here also you can see that after 30 days, there is a significant fold change or high upregulation of the microRNA post-infection in both spleen and liver. So this upper panel suggests that the microRNA is not only getting upregulated in the BMDMs or the in vitro macrophage system, it also got enriched in infected animal and different organs of the infected animal in a time-dependent manner. After that, we wanted to know that whether this microRNA has anything to do with entry of the parasite. So as I mentioned earlier, that when the parasite is entering in the human blood, it basically is engulfed by the macrophages. And what is the mechanism of this engulfment is basically the macrophage eats the parasite, and the method we call as phagocytosis. So in plate or in vitro, if you want to study the uptake of the amount of uptake or the phagocytosis, the method we use is basically we label the parasites with a green fluorescent dye, which is called CFSC. And after labeling those parasites, we use those labeled parasite to infect macrophages. 4 hours post-infection, we remove or wash out the extracellular parasites, and then we also fluorescently label the macrophages with a different color of color. Like here I have used PE labeled CD11B, which is a red color fluorescent dye. And from there we can use flow cytometry method to understand that what is the percent positive CD11B positive cells that has green signal or CFSC labeled signals inside them. So the changes that we tried to make here is we used one set of macrophages, which is normal, like wild type, and we used another set of macrophage where we tried to inhibit the mature forms of the microRNA using a synthetic inhibitor. So we bought that inhibitor from market, and we used that inhibitor, or we lipofected the inhibitors inside the macrophages. So here our main goal was to understand that if we delete or if we remove the mature form of the microRNA, whether it hinders or it helps, like it has positive or negative role on the uptake of the parasite in CD11B positive cells. And why I chose this CD11B, so it is a pan marker for macrophage. So if you if you want to catch the macrophages or not the monocytes, you can use CD11B as a marker. So this is the representation of this is the dot plot and histogram representation of the mean fluorescent intensity or MFI. So as I said that this CD11B positive cells which I tried to gate by the circle, and these cells are those which has CFSC inside them. So CFSC is nothing but the parasite, which represents the parasite inside these macrophages. And in this panel, the histogram panel, it represents the mean fluorescent intensity or the intensity of the CFSC inside these CD11B cells. So you can see that in normal macrophages, which is infected with the parasite, the MFI was around 500. And then we treated the macrophages with inhibitor, that is anti-146A, you can see there is a sharp decrease of mean fluorescent intensity from 510 to 40. Now, what is this middle group? that is anti-NC. So whenever we are trying or we use any kind of inhibitor RNA or any kind of siRNA, always we have to use a control RNA, which will which will remove any kind of off-target scoring that we do. So here we used a random oligonucleotide, and that is we mentioned as anti-NC. So this also showed that there is not so significant decrease of MFI. So in anti-NC treated BMDMs, when we infected them with CFSC level parasite, we can see that the MFI dropped from 510 to 450. So from this data, we can say that whatever change of MFI or the parasite uptake that happened in the macrophage is happening due to inhibition of microRNA 146A. And similarly, we also took some of the cells out and then we performed. So if I just go back to this slide. So this is the CFSC labeled parasites which we used to infect the BMDM to do this flow study. And on the other hand, we used normal parasites like unlabeled parasites. And from there, we took the infected macrophage 24-hour post-infection, the normal method or normal way we use for infecting any macrophage. And 24-hour post-infection, we stained them with a standard staining method, that is GIMSA stain. And from there, as I mentioned earlier, from there we counted the number of amastigotes per 100 macrophage. And we plotted them in the right panel, which I mentioned as macrophage parasite burden. So you can see that not only my flow cytometry data, but also this GIMSA stain data suggest that per 100 BMDM, the number of amastigote dropped significantly in MIR146A inhibited group compared to healthy, infected. Sorry.
Sorry for the disturbance, ma'am. I am just muting.
Okay. It's fine. Yeah. So both of the. Yes. Thank you. So both of these studies indicated that the parasite uptake decreases when you inhibit this particular microRNA. So the microRNA has something to do with the uptake or the phagocytosis of the parasite inside the macrophages. So next, what my next aim was to relate or find out the correlation between macrophage plasticity and infection burden when we and also with the microRNA fold change. So here I have represented fluorescent microscopy fields of 4 different types of macrophages. So the upper left panel is uninfected BMDMs. And then when we infect the BMDMs, without changing anything, the BMDMs are normally layer plated and we do not give any kind of stimulation to make them either M1 or M2 type. So this type of BMDMs will be mentioned as M0 or naive BMDMs. So when we infect naive or M0 BMDM, here I don't know how far you can see these small blue dots surrounding this big blue circles. So these small blue dots, they indicates the number of amastigotes. So I tried to point out some of the bright points, if you can see. And then from there, what we did, we polarized these M0 macrophages. So in another set of experiment, we polarized this M0 or the healthy macrophages to M1 type. So how do we do that? We generally use LPS and IFN gamma together to polarize the M0 to M1 type. And in this M1 type of macrophages, we gave the infection. And on the other hand, we polarize the macrophages to M2 type by giving IL-4 and IL-13, and then also we gave the infection. So you can see by your eyes that the number of the blue dots surrounding this macrophage nuclei increased dramatically when they are polarized to M2 type. So this means that the if you synthetically or if you if you change the macrophages initially from M0 to M2 type and then you infect them, it helps the parasite proliferate so nicely. So the macrophage polarity from M1 to or the M0 to M2 type has a definitive positive correlation with the parasite burden. On the other hand, if you look at the M1 type, compared to M0 and M2, the number of blue dots in the M1 macrophage is significantly lower. So it says that compared to M0 or M2, the survival rates of the amastigotes inside the macrophage is pretty low. So this data indicated that M2 type of macrophage helps or allows more proliferation of the parasite compared to M1 types. So our next goal was to understand if you make if you infect the macrophages with the parasites and also inhibit the microRNA using the same inhibitor that I used before for doing my flow cytometry data. What is the outcome on the so the outcome we looked into was based on the what is the marker expressions for M1 and M2 type. So far, our story suggested that this Leishmania infection helps M2 type of macrophage, or M2 type of macrophage helps Leishmania infection. So this two biological event has a positive correlation. And there comes a third player, that is microRNA 146A. So if you delete that third player and infect the macrophages, what will happen? So if this microRNA helps the parasite to make M2 type of macrophage. So the microRNA, when the microRNA will be deleted, the whole M2 type of profile should move towards M1 type, even if there is infection. So if this happens, then we can say that yes, this polarization of macrophage towards M2 type by the parasite is happening via microRNA 146A. So to find out that question, or to answer the question, we looked up into some specific markers of M1 and M2 type. So you can see that the markers for M1 type include some transcription factor, that is phosphostat1, IRF1, AP1. That includes the main target proteins that I showed in my previous slide, that are target pathways of the target protein of the microRNA. As we are inhibiting the microRNA, so we will definitely look into the profile of those targets. So we will be looking into IRAC1 and FRAT6. And these target or the proteins are the part of NF-kappa B signaling. So we will be looking into P65, which is the main subunit of the NF-kappa B pathway. And then also we will be looking into other cytokine markers and mediators of M1 like IL-12, TNF-alpha, INOS, that is induced nitric oxides and nitric oxide synthase. And in part of the M2, we will be looking into transcription factors that is important for making M2 like phosphostat6 and CEBP beta, and the cytokine IL-10 and arginase 1. So what we will do, we will inhibit the microRNA and then infect the macrophages with parasites, and then we'll perform Western blots. We have performed Western blots and also nuclear translocation of the NF-kappa B P65 subunits, and we have done some flow and ELISA based study to understand cytokine profile. So I will not show all of the data. So I'm just highlighting some of the major findings that I got after this experiment. So here you can see the profiling of the transcription factors. So the change in the transcription factor mainly represents that the cells are finally, or for the definite stage, they are transformed. So transcription factors are the end stage of transformation in either M1 or M2 type. So here you can see that these are the M1 transcription factors. They are significantly upregulated or enriched after inhibiting this microRNA. So compared to this control treated group, you can see that compared to uninfected group, this control treated group has significant or significantly lower amount of this particular marker proteins or transcription factors. But when we are inhibiting this microRNA and then infecting, we can see a significant enrichment of all of these transcription factors, which indicates M1 type of profile of those macrophages. On the contrary, if we look into this M2 type of transcription factors, we can see that compared to anti-NC treated group, there is a significant decrease of p-STAT6 and CEBP beta in anti-146A treated group. So this denotes that inhibition of microRNA is preventing the formation of AIM2 type of macrophage by LD, like Leishmania donovani infection. And then we also saw that as we are inhibiting the microRNA, so its target proteins like TRAF6, IRAC, they are significantly enhanced compared to anti-NC group. And the downstream mediators, that is INOS, is also enhanced, and arginase 1 is decreased. So this, as I mentioned earlier, that INOS is another marker of M1 type of macrophage, and arginase is a marker of M2 type of macrophage. So in correlation with my previous slide, where we found that transcription factors for M2 got significantly downregulated after inhibiting the microRNA, we also saw that arginase 1 is also downregulated. And similarly, like the M1 type of transcription factors, they are upregulated. M1 mediator INOS or NOS is also upregulated. So the whole story of the whole Western blot data suggested that inhibition of microRNA is preventing formation of M2 type of macrophages. Now, after that, we did this nuclear translocation assay by using both Western blot and also confocal based co-localization experiments to understand that whether this P65 or the nuclear, new NF kappa B is activated and nuclear translocated inside the nucleus when we are inhibiting the macrophage. Why we are looking into this? Because we are inhibiting the microRNA, which targets TRAF6 IRAC1. So if you inhibit this microRNA, TRAF6 and IRAC1 will be present in the cell, and they will finally help this P65 subunit to get phosphorylated and translocated in the nucleus, and there it can bind with different kinds of pro-inflammatory cytokine promoters and also nitric oxide promoters, and help to produce the further their expressions. So you can clearly see from this microscopy images that compared to this anti-NC group, 146A inhibited groups has higher amount of the combined signal from green and blue. So if you look into this graph, which indicates the combined signal from the percent of combined signal from green and blue, it is the amount that of the combined signal is represented as co-localization coefficient. So it is around 0.78. So it is 0.35 when we are infecting healthy BMDMs. So we can say that if this coefficient is more than 0.5, we take it as a positive coefficient or positive co-localization. So when we are inhibiting the microRNA, you can see this co-localization is very much positive, and this indicates that most of this phosphorylated P65 is translocated in the nucleus. And we also supported this data by isolating nuclear fractions and cytosolic fractions from all of these groups. And we can see that compared to anti-NC treated group, there is a significant amount of phosphorylated P65 is translocated in the nucleus. Next, we studied some major cytokines which will represent the polarization status of the macrophages after inhibiting the microRNA. So we found that after inhibiting M146A, there is a significant increase of IL-12 and TNF alpha, and there is a downregulation of IL-10, which is M2 polarization macrophage, M2 polarization cytokine. So I apologize for this mistake. So here, just by mistake, this IL-10 popped up on this purple bar. So here, this pink bar represents IL-12. This purple bar represents TNF alpha, and this red bar represents IL-10. So here you can see this colorful bars are mainly representing the flow cytometry plot that I showed in the top panel, and the black bars are representing ELISA data. So we perform the flow cytometry experiments to understand the expression of the cytokine, and followed by that we did cytokine ELISA. So both of the studies or both of the experiments suggested that when we inhibit the microRNA, secretion of M1 type of cytokines, that is IL-12 and TNF alpha, enhanced, and M2 type cytokine IL-10 decreased. So far, what we learned is that basically when we infect the mice or the macrophages, we end up getting higher amount of the microRNA. And when we are inhibiting, and this microRNA helps polarization of net naive macrophage or M0 macrophage towards, mostly towards M2 type. When we are inhibiting this microRNA, we end up getting less amount of infection burden, higher amount of M1 markers like the marker protein, cytokine, and also nuclear translocation of P65. Then we wanted to understand what this is, the in vitro system, right? So we wanted to understand what happens if you inhibit this microRNA in vivo and then infect those animals with the parasite, and then study some of the major markers of the polarization and also parasite burden and the TRAF6, IRAC1, this kind of thing. So what happens when the whole story, we try to look into the whole story of the signaling pathway after inhibiting the microRNA in animals. So this is a first kind of report where we try to inhibit or use a particular type of chemically modified antisense oligos or ASO to inhibit microRNA in vivo. So what is the specialty of this ASO is it has very high affinity compared to standard inhibitors that is available in market. It has significant resistance towards nucleases, which is very important because when you are trying to put some kind of RNA or oligos through blood, it is very important to make them stable in presence of different kinds of nucleases, which is present in our blood, and it should have very good retention in vivo. So this type of ASOs, which has a special kind of modification 2' O methoxyethyl in methoxyethyl, so it helps a very good in vivo retention, because we are going to do this study for very long days. So we need to make sure that the microRNA inhibitor stays so long. So for these reasons, we use this kind of modification, which mostly is done in the 2' part of this microRNA, and this is made from thermo, which is also sold as Nirvana inhibitors. So what we did, we infected the mice via tailwind route using this particular type of inhibitor. Here also we have used non-specific oligos as our control. So we injected one group of mice with PBS, one group of mice with anti-NC or the control oligos, another group of mice with Nirvana inhibitors. And 28 days after infection, along with this after injecting this ASO, then we infected the mice, and at the 35th day after infection, like 7 days after infection, we tried to sacrifice and then look into the amount of the parasite in mostly the spleen and liver. So what we saw that this is the LDU or Leishman-Donovan unit. So it is nothing but standard gene sustaining method, which I mentioned many times earlier. So what we did, we isolated this spleen and liver and then made slides of their sections, which we call as stamp smears, and then stain those slides using gene sustain. And from there, you can simply use light microscope to count the number of amastigotes in the organ. And here the amastigotes are represented, number of amastigotes in spleen and liver are represented as LDU. So you can see that compared to anti-NC treated group in both and spleen and liver, after inhibition with this ASO, there is a drop of parasite burden, which is significant. And after that, as we are using this microRNA inhibitor, so it is obvious that we will look into the expression of the microRNA. So here I have represented the expression panel which we found from spleen. I also looked into the liver as well, liver expression profile as well. So you can see that the inhibitor is really quite stable, and it has a very good capacity to inhibit mature microRNA expression compared to the anti-NC treated group. So in the left bar graph, you can see that there is a significant drop of fold change or FC microRNA 146A in anti-146A treated spleen compared to anti-NC treated spleen. And further, we looked into the major markers of M1 and M2. As I mentioned earlier, that INOS is a one type of this standard marker for M1, and arginase 1 is a marker for M2. So we prepared tissue lysates from spleen and liver from PBS, anti-NC, and anti-46A treated groups. And we performed Western blot, and we saw that after inhibition of the microRNA, there is significant increase of INOS expression and downregulation of arginase 1 expression in compared to anti-NC treated group. After that, we looked into some of the very well-known M2 polarization markers, which is YF1, FIS1, CCR7, and arginase 1, which I also did this Western blot as well. So you can see that in both spleen and liver, there is decrease of all of these M2 markers compared to anti-NC treated groups. And then we performed cytokine ELISA from the splenic or the splenocyte cultures that so what we did, we isolated the splenocytes from the spleen of all the groups, and then cultured them in presence of leishmanial antigen or lag. So you all know that when we are trying to stimulate, like when we are trying to culture any infected organ samples or organ cells in media, you have to keep on giving some kind of memory response of the same infection. Else the organ cells will forget the whatever infection background they had, and they will not give you the ideal profile you are expecting. So we isolated these organs from the animals which are infected with Leishmania donovani. So when we are culturing the organs in plate or culture media, we have to provide leishmanial antigen as a memory stimulation, so that the organ macrophages will keep on secreting all the necessary cytokines that will come out of the memory response. So we looked into standard like IL-10 and IL-12. So IL-10 is representative of M2 cytokine, and IL-12 is representative of M1 cytokine. So you can see that compared to anti-146A groups, compared to anti-NC treated group, when we inhibited the microRNA, there is decrease of IL-10 and increase of IL-12 titer in splenocytes. So here this total so far the story suggested that not only in cells like BMDMs, if you inhibit the microRNA in animals and then infect them, we end up getting low parasite burden. And also the data I didn't show here, so we saw a very good size regression of spleen and liver. As I mentioned earlier, that if you infect mice or human with this, like when the humans are infected and also the mice, we infect them with parasite, it gives a bigger size of spleen, which we call as splenomegaly, and the liver size also increases, which is as hepatomegaly. So when we inhibited the microRNA and sacrificed the animal compared to the control groups, we saw healthy size or the smaller size of the spleen or liver.
Chalo, let's go. Let us go. Let us go.
Sorry for disturbance, ma'am.
All
Yes, ma'am, you can continue.
And
Dear participant, please be unmuted during the session. After that, you can unmute yourself and ask your query. So please be muted. Yes, Sonali ma'am, you can continue.
Okay, thank you. So next we try to understand how infection triggers this microRNA. So for that, we looked into a very interesting type of transcriptional regulators, which is recent days they are coming up. They are known as super enhancers. So what we wanted to do, so this is kind of a first in case report where we studied this kind of elements or DNA elements and their role in infection model, like mostly in the Leishmania infection model. And our group for the first time identified that microRNA can be regulated by this kind of elements like super enhancers. So what are the super enhancers? So we all know that the transcription of any gene is transcribed from the location which we called as transcription start site or TSS. So when RNA polymerase binds the upstream region of TSS or the promoter, then it helps transcribing the RNA from the TSS. Now there are more elements which is present at the
Ma'am is not audible, sir.
Sorry, but you are muted, sir.
Yes, sir. I think she lost the connection. So I'm contacting.
Hello.
Yes.
Sir, is there any feedback form or exam?
Yes. The feedback form will be there.
Okay, actually, I didn't get right now.
No, we have not shared it.
Okay, okay. And you told that you will take one exam also.
Yes.
So when it will be tomorrow or today?
Yes, I will share in the group.
Okay, okay.
Hi, Dr. Kumar.
Hello, ma'am.
Yeah, so I think I was at the last slide of my presentation,
Yeah,
so I
yes.
can just share that.
Yes, yes, please.
Okay, so far, so this is the total whatever finding I showed is that if we are trying to search some promising therapies for Leishmania donovani, because like visceral leishmaniasis, because it is a very prominent and endemic disease of India and as well as in the different kinds of parts of world. So my approach was to understand different kinds of host factors. So mostly here the host factor was microRNA, and screen them out and then find out the different kinds of controllers of the microRNA and how does the microRNA helps in manipulating macrophage polarity. So inhibiting the microRNA both in macrophages and mice helped me to prevent the outcomes of the disease and make it more like in a better condition. So we are proposing by our work, we proposed or try to propose a process of future immunotherapy by using different kinds of RNA inhibitors in macrophages, and that can be a positive, possibly that can be a promising future therapy, therapeutic approaches. Okay, so this is the lab group where I worked in India. So I worked with Professor Nahid Ali. She is a J.C. Bose National Fellow in CSR ISAP Kolkata. My work was funded by GCRF from UK and also CSR and NSERB. Thank you everyone for listening and for your patience.
Thank you. Thank you so much, ma'am. So I would like to extend my heartfelt gratitude for your insightful and thoughtful provoking talk on the role of mRNA in regulating macrophage plasticity in leishmaniasis. So, ma'am, your expertise in this field is truly remarkable, and your presentation shed light on the complex aspect of the disease with clarity and depth. And your dedication to advancing our understanding of leishmaniasis and its mechanism is highly inspiring. And I am highly grateful for this opportunity to learn from your expertise. And thank you once again for sharing your knowledge and insight with us. So, dear participant, if you have any query, you can unmute yourself and ask your query. Do you have any query related to Kala-Jar, leishmaniasis, or any infectious disease? You can ask. Okay, Sanjay sir is asking that which gene in leishmania parasite decide the occurrence of visceral or cutaneous leishmaniasis?
Okay, that's an interesting but very complicated question because there are, yeah, because normally it is not so well known that is there any special kind of gene which is dedicated to like from the parasite part. So the difference between visceral and cutaneous leishmaniasis is mostly based on where the parasite is infecting. So in case of cutaneous leishmaniasis, the parasite infects mostly in the skin part and it resides and it gets remain there over there and it makes all kind of damages in the skin, nose, mouth, this kind of parts. But on the other hand, in case of visceral leishmaniasis, the parasite mostly enters to the in the spleen, liver. So it never stays on the skin, and also their species are different. So their total lifestyle, life story, and life cycle goes on in the internal organs. So I will not be able to particularly tell you that what are the proteins or antigens because there are so many similarities between the two type of parasites which causes VL and also CL. But yeah, I can definitely tell you the location of the host macrophage or host body where parts where the parasite is infecting has a lot to do with its whether it is a visceral or a cutaneous ones.
Okay, any other query? You can unmute yourself also and ask your query. So Priyanshu sir is writing a very informative session, and thank you, ma'am. Again,
Thank you so
Sanjay
much.
sir, Sanjay sir again get a satisfactory answer, and thank you to you. Okay, any other query? I think there is no query. So on the behalf of organizing committee, ma'am, I am highly grateful to offer you as a e-memento as a part for being a part of this 5 days online faculty development program where more than 200 participants has attended this event from more than 20 states. So kindly accept our this honor and gratitude from the Galgoti University, ma'am.
Thank you so much. Thank you.
Thank you so much. Okay, dear participant, I have sent the feedback form link in this chat box. Please provide your feedback